Welcome to A Fresh Face in Hell. I'm your host, Jonathan Evison. Every week we talk to someone new and interesting, somebody intellectually curious, somebody who will hopefully renew our faith in this cold, cruel world we live in. A Fresh Face in Hell. Today, we are going to talk about genetic engineering. Specifically, we're going to talk about something called CRISPR. Now, what exactly is CRISPR? It sounds like an app, but in reality, CRISPR is the biggest breakthrough in the history of genetic engineering. There's a guy out there who's trying to bring the woolly mammoth back to life using CRISPR. CRISPR is something that can eradicate diseases potentially. They've had great results with sickle cell anemia, but who knows, maybe cancer. There also comes the, designer babies. Anybody? Today, we're going to talk to Kevin Davies, who's the author of Editing Humanity, the CRISPR Revolution, and the new era of genome editing. Like I said, this is heady stuff. I'm not much of a biologist, but Kevin's got a PhD in genetics from Oxford, so I think he's capable to walk us through this in layman's terms. As I said, the possibilities are really mind-blowing here, so stick around. But they're also quite scary, and it's rife with, ethical problems as well. So we're going to have to dig into that. Frankly, I can't believe we're not all talking about CRISPR because it's kind of mind-blowing. So, hopefully today on A Fresh Face in Hell, we're going to put CRISPR on your radar. Please welcome to the show Kevin Davies, who joins us today from Washington, D.C. So, Kevin, thanks for joining us.
My pleasure. Great to be here.
Yeah. So, I'm not the sharpest tool in the shed when it comes to biology necessarily. I'm more of a cosmology guy. CRISPR's been on Jason, my producer's map for about 6 or 7 years. So he's been briefing me on this. But for the sake of the listeners and for me, I'm going to ask you to be patient, and we're going to kind of need to start from the beginning with CRISPR, which sounds like a dating app to me. Tell us, what is CRISPR?
In modern usage, CRISPR is a tool that we now, scientists around the world, use to edit DNA. We can edit pretty much the DNA of any organism. There are many flavors of CRISPR, and we can now make the most precise, finessed sequence changes to the genetic code of any organism. Obviously, humans come to mind as one of the most important organisms that we might want to potentially use CRISPR on to reverse hereditary mutations, for example, to potentially cure cancer. But we can use it on a whole menagerie of other organisms, animals, plants, microbes, and I'm sure we'll get into some of those applications as we go on. CRISPR, however, was not invented in a lab per se. It's a naturally occurring phenomenon, but was only discovered about 20 years ago. It's at the root of it. CRISPR describes a bacterial immune system, and many of your podcast listeners are probably thinking, why would bacteria have an immune system? But bacteria, like human beings, are prey to viruses. And they have evolved over millions, if not billions of years of evolution, their own systems, their own layers of defense to detect and fend off viruses that would otherwise invade the bacterial cell and destroy it in the course of the viruses replicating. CRISPR is one of these sort of programmed anti-missile defense systems that bacteria have evolved, and scientists sort of stumbled onto this about 20 years ago and realized that bacteria had this beautiful system to recognize specific sequences of viruses and then deploy a kind of an enzyme that acts like a scissors to cut the DNA of those invading viruses. And what scientists like Jennifer Doudna, who went on to win the Nobel Prize, realized about 12, 13 years ago was, if we just made a couple of subtle tweaks to the machinery that's involved in this immune system, we could now take that system and deploy it in a test tube or in human cells or the cells of any organism. And now we finally have in our toolkit, in our genetic toolkit, a system to make a meticulous programmed specific changes to the DNA. And of course, that just unleashed a whole host of applications and diseases that we might want to tackle. And it's been a very exciting decade in molecular biology.
Yeah, so I mean, CRISPR basically invented itself. We didn't, it's with a name like CRISPR, it sounds like some sort of technology we have but we have adapted it. But like, I mean, I'm still a little, I mean, is it a protein? Is it just a genome in the sequence? Is it?
It's a bit of great question. It's a bit of both. It's a basically a combination of a protein, an enzyme, and a specific kind of enzyme that we call a nuclease. So a nuclease cuts genetic material, DNA and RNA. So that's really a very important key component of the CRISPR system is an enzyme that bacteria produce. They have encoded these enzymes encoded in their own genome. Now, you don't want a nuclease just running around a bacterial cell cutting DNA at random. Otherwise, the bacteria wouldn't live very long, right? So the enzyme by itself doesn't do anything, but it needs a guide. It needs a GPS signal, and the GPS signal is provided by a tiny motif of RNA, which is the chemical cousin of DNA. Not everyone, but many of your listeners have received RNA vaccines during the COVID pandemic. And so this little RNA signal provides a specific sequence that tells the enzyme, you go cut anytime you find a stretch of DNA that contains the matching sequence to this GPS signal that you're carrying. And where does that GPS signal come from? It comes from the remnants of a virus that the bacterial ancestors encountered, said, you're an enemy, you're a foe. I'm going to hold on to a little piece of your DNA and stitch it into my own DNA. And that will be my cue card, like a mugshot that I can look up. And if I encounter another virus and I see that you match the mugshot, then I know I need to put you away. And so the CRISPR editing system comes from a combination of the nuclease with this GPS signal. And what scientists like Doudna and her colleague Emmanuelle Charpentier figured out 12, 13 years ago was if we just play around with that GPS signal, in principle, whatever we can retype that GPS signal to target any gene that we might want to go into and cut. And the breakthrough papers that led to the Nobel Prize were published in 2012, and a whole bunch followed in 2013 that proved that yes, we can adapt this naturally occurring CRISPR system, and now we can pretty much program the sequence. There are a few caveats, but we can pretty much program any specific gene sequence. And now this CRISPR will go cut. And once it cuts, then a whole bunch of different things can happen. But the end game is that we can use this system to make now very specific changes to the DNA sequence. And from there, it's just the limitation of the imagination of the scientists that really is the only thing holding us back.
I mean, like, this could either be our savior or it could undo us. And I mean, as we go on, I still want everybody to get a kind of a scientific understanding. You can see I'm probably going cross-eyed during some of this, but I mean, the ethical concerns alone, we'll start to tackle those. But I'm going to back up. Basically, nature is so smart. Who knew bacteria was so smart? So basically, somebody finds this in a petri dish or whatever and says, "Hey, what's that sequence there?" Right? What is this? What is the bacteria doing? What does the bacteria know that we don't know? And then we learn from it.
Yeah, that's exactly how it began. The CRISPR saga began in the late 1980s when a Japanese group discovered a very weird section of DNA in a bacterial genome that they were trying to sequence from start to finish. And it was a curiosity. It reminds me of, those crop circles, those aerial photos that you see in the countryside. And you think, that is bizarre. That can't have gotten there by accident. Somebody put them there, and they must have some sort of purpose or function, religious or whatever. And when these scientists discovered this strange repeat motifs in the DNA. They said, well, that's weird. That must be doing something. DNA doesn't assemble in these strange patterns just at random. But that wasn't the problem they were trying to solve. So they noted it in their paper and moved on. And over the course of the 90s and early 2000s, just a handful of microbiologists were really intrigued by this observation, and they started studying it. And I think the most famous of these guys was a Spanish microbiologist named Francisco Mojica, who really became quite obsessed with what is going on with these repeat blocks of DNA. And they came up with the name clustered regularly interspaced short palindromic repeats. So CRISPR is an acronym that actually stands for something, but we don't need to dwell on that. And what intrigued him wasn't so much these repeated stretches of DNA, but sandwiched in between those repeats was other rather short DNA fragments that he felt had to be doing something or must have some functional significance. So he would go into his lab every day and type in some of these sequences into the computer to search against the database of all known DNA sequences. And for months, if not years, he kept drawing a blank. Nothing was coming up. And suddenly one day he typed in one of these particular sequences, and boom, the computer spit back, this matches a virus. And over the course of a very short space of time, scientists, including Mojica and others, realized, why would bacteria be stitching into their own DNA sections of viral DNA? And before long, it was clear that this was the immune system. And a couple of years later, a guy named Rodolphe Barangu, who's a professor at North Carolina State University, with whom I work, we launched the CRISPR Journal together about 7 years ago. He was the first to experimentally show that if you match the right CRISPR sequence with the right virus, then that virus will be no more, because the CRISPR is providing the GPS signal to the nuclease we talked about a minute ago, and that's destroying the virus that would otherwise infect and wipe out those bacterial cultures.
So it's really, in essence, it's kind of like the human, it's kind of like our immune system in that it contracts something and saves the information to, like a shot works, where we actually contract it and then our body saves that information. But we're just talking about this on just a tiny cellular level.
Right.
And like so much, I mean, it's like a cliche in science that accident. I mean, so many of the great scientific discoveries are discovered by accident. This I'm going to just call him Mojito.
Mojica with a C, yes.
Mojica. So this guy basically, I mean, so once you can isolate this sequence, understand it is, and then start to switch it out, you can basically create. Is that what the gene drive is? Is that what we talk about when we talk about?
Gene drive it's probably good to, just before we get into gene drives, just say that once we had in 2012, 2013, 2014, the fundamental description of this new gene editing system. CRISPR was not the first gene editing technology to be developed, but the ones that had come before were very hard to work with. Only a couple of very specialized companies with dozens of scientists really trying to fixate on this one particular issue were able to get these things to work. The beauty of CRISPR was that any scientist, even with only a paltry amount of real molecular biology training, could get the ingredients, put them in a test tube, and start running CRISPR experiments. Pretty soon, high schools and undergraduate courses in colleges were running rudimentary CRISPR classes. And CRISPR, you didn't need expensive machinery or equipment. You didn't need some fancy $1 million DNA sequencer to get this to work. So labs around the world literally were able to jump onto this bandwagon and use their imagination. So by 2014, a whole bunch of biotech companies were growing up, mostly in Boston and San Francisco, the usual suspects. And these were after, okay, if we can edit DNA now, the obvious thing we want to do is to tackle genetic diseases like sickle cell disease, like liver diseases, maybe down the road, Alzheimer's disease, and other whole host of, there's thousands of genetic diseases that we might want to tackle. We know the DNA mutations that cause these diseases. Some are a bit more complicated than others, but for thousands, we know exactly what the cause is. And for 20 or 30 years, we've been trying to treat many of these patients with these sorts of diseases using gene therapy. And when I talk about gene therapy, we typically mean just take a healthy copy of the gene that's mutated, add it to the cell, the liver cell, the brain cell, the muscle cell, the lung cell, whatever the cell that's affected in that disease is. And hopefully the healthy gene will kind of outcompete the broken gene, the mutated gene that you've inherited, and then all will be well. Well, it's just not that easy. Biology isn't quite that easy. And the viruses that you use to shuttle the healthy gene into the cell, well, they can cause a whole host of other unwanted problems that led over the course of several decades to a number of patient deaths and set back the gene therapy field, several times. Gene editing offered a much more precise and subtle way of doing this. Why add a whole gene when if you could just add the molecular machinery, the scalpel, if you will, to perform what one European scientist in this field called DNA surgery, then why wouldn't you do that? What a brilliant way. Just imagine a little scalpel floating into the nucleus of a cell and performing that sort of keyhole surgery literally on your DNA. And that's really what
So like gene
CRISPR does.
therapy is just like throwing more infantrymen down there and hopefully they can, whereas this is more like we've got intel.
That's
We're,
right.
okay. So yeah, it's just, okay. So most of the work now and is still kind of going on in the lab. I mean, what are the biggest challenges standing in the way of the like widespread clinical use?
Well, it is, you say yes, of course, there's a ton of work going on in the lab. But what's been really exciting over the last 5 years now is seeing the progress that CRISPR has made in the clinic. And indeed, this has really inspired me to write the new book that I'm working on, which is going to be a biography of sickle cell disease. So in the summer of 2019, National Public Radio put out an interview with an African-American woman who was the first American woman to volunteer in a CRISPR gene editing clinical trial. She has sickle cell disease, which is a terrible disease that affects about 100,000 Americans, mostly people of color, because the mutation literally came from Africa during the Middle Passage. It arose in Africa tens of thousands of years ago. And for Victoria and hundreds of thousands of people like her, a sickle cell was a daily, if not weekly, survival course of dealing with the most excruciating pain episodes, frequently brought on by either exercise or dehydration or cold weather, or they could just come out of nowhere. No cure. The pharma industry has done a pretty lousy job of prioritizing and doing anything for these patients. But CRISPR provided a very clever workaround to potentially treat sickle cell disease. So we know that quick bio 101 on sickle cell. This is a disease that affects hemoglobin. That, of course, is the crucial protein in all of our red blood cells, the most common cell type in the human body. And hemoglobin is made up of two chains, an alpha chain and a beta chain. It's the beta chain that contains a single mutation that causes sickle cell disease. So if you're trying to treat sickle cell, you're left with two choices. One, either try to go in and directly fix the mutation. But there's another workaround, and that is before we're born, our hemoglobin, while we're fetuses, if that's the right plural term, is made up of alpha and gamma, so a different chain. And for decades we've known if we could just find the gamma gene in the genome and turn it back on, that would replace the faulty beta chain, and then we'd be able to potentially repair hemoglobin and give these patients a fully functional hemoglobin.
Wow, so
Victoria,
that's in real time.
yeah.
That's for the patient, not just genetically going forward, we're gonna cut
Oh,
sickle
this
cell.
is one patient at a
So this
time.
is a treatment. Yeah.
Correct. So Victoria became patient number one in this trial that was launched by a biotech company called CRISPR Therapeutics and then taken over by Vertex Pharmaceuticals. Fast forward 5 years, she and dozens of other patients are effectively cured. and I really hesitate to say the C word, but in this case, it's proven true. It's proven in their daily lives. It's proven in peer-reviewed publications in the leading medical journals, and is proven by the auspices of the FDA, which approved this therapy in December 2023. And now the first patients in hospitals here in DC and elsewhere are finally getting insurance coverage so that they can take these therapy. This therapy manages to switch back on this gamma globin chain, and that just completely transforms the production of the blood cells in these patients. It's not an easy procedure. You have to, it's basically a modified bone marrow transplant. The patients have to undergo chemotherapy. The chemotherapy, it takes place after they've harvested their own stem cells from the bone marrow. So that's one process. Then they take chemotherapy to clear out the bone marrow to provide purchase for the gene-edited stem cells months later to be infused back into the patients and take root, and then they start pumping out the trillions of red blood cells that circulate in our body, 24/7. But Victoria is who I interviewed for my new book, and many other patients. They're doing fantastically well. They have no more pain crises by and large, no more hospitalizations, no more need for blood transfusions. You could not have dreamed up a better scenario for CRISPR or for any type of genetic medicine really than this story. So that's the good news. The bad news is there's a price tag, not just the one that the patients have to spend sort of metaphorically that I just described. There's a literal price tag of $2.2 million for the therapy on top of the hospital stays and bone marrow services and so on that the clinics provide. But insurance companies are beginning to cover this because they do the math and they say, yeah, $2 million, that's a heck of a lot of money. But the lifetime cost of all these blood transfusions and complications and organ damage and operations, top that up over 10, 20, 30 years, and suddenly $2 million looks like a bargain, particularly if the patient never, or I don't want to say never, knock on wood, but seldom has to go back into hospital. So that's the economic bargain that we're seeing out. And that, for the time being, is the poster child for CRISPR medicines.
The sickle cell. And of course, with the insurance, it's always got to be about the numbers, not about the humanity or when you're talking about inside the bones and using sickle cell was a great target for this therapy. I can't help. I have a very dear friend with Duchenne muscular dystrophy. And as I understand it, the bone inside the bone marrow is where dystrophin is created. And this is what people with Duchenne muscular dystrophy don't have. Their body doesn't produce dystrophin. So their body basically just goes into that rapid decline in terms of, can we produce dystrophin with this?
Certainly there are companies that are trying to target muscular dystrophy. So dystrophin is a muscle protein. So it's the muscles that we have to address in the case of that genetic disease, which is one that I worked on briefly as a grad student. So that one's quite near and dear to my heart. One of the problems with tackling a disease like muscular dystrophy is we have lots of muscles. There's lots of cells that would have to be repaired in order for the patient to feel manifestly healthier and stronger and more mobile. So that means you've got to get more gene therapy or more CRISPR into those cells. And when you start upping the amount of the titer of virus that is used to transmit the gene or the CRISPR machinery into those cells, then the dangers of that start to ramp up. There was a gene therapy trial, fatality reported quite recently in a gene therapy trial, not CRISPR. That is, the trial was being sponsored by a biotech company called Sarepta, which has otherwise reported great success in this trial, treating hundreds of children with Duchenne muscular dystrophy. So one adverse event out of hundreds is still a very good record, which is not to diminish the tragedy of losing this one particular patient. But so there is hope for that, and hope for a slew of other disorders. The next disorders that will likely be the next trials that will be presented to the FDA for approval are a couple of liver diseases where we've found companies like Intellia Therapeutics have found great success in getting the CRISPR enzymes taken up by the liver, because the livers are very effective sponges, so they like doing that. And they've been able to do this by directly injecting that CRISPR into the patient in a not a virus this time, but something called a lipid nanoparticle, a very inert ball of fat in which you can encase the CRISPR, and that gets taken up by the liver cells. That seems to be working really well. Those are much rarer disorders where we're trying to stop the toxic buildup of certain proteins. But I think based on the clinical results reported so far, we should soon, probably later this year, hopefully, have another couple of genetic diseases now amenable to CRISPR therapies.
Cool. So now the technology is new, particularly in the clinical environment. So we don't really have any long-term view of the results. I mean, should we be concerned about the possibility of unintended consequences? I mean, I believe you call them off-target effects with CRISPR. I mean, could you offer some example of what some off-target results might be?
So we definitely should be vigilant to these, and I commend the FDA and all the sponsoring companies because the last thing they want is to be presiding or approving a clinical trial and then say, well, we didn't anticipate this potential problem. And now we're paying the price because we're seeing unwanted consequences. So before the sickle cell therapy was approved, the FDA held an all-day panel in which the company presented to really just dig into what are the risks and how do we ensure and follow the patients to make sure this doesn't happen. So all of the patients who are in this trial will be followed for another 10, 12, 15 years. And the moment anything untoward pops up, you can be sure that this will be disseminated across the community. The big concern, there's obviously with any new drug, there's going to be some safety issues that have to be addressed and monitored. The concern with CRISPR is that you're using a GPS signal, right, to target a specific sequence, and the DNA sequence is made up of these 4 chemical subunits that we abbreviate A, C, T, and G. So that's the genetic code. So CRISPR's looking for a specific string of A's, C's, T's, and G's in a specific gene, and that's where the CRISPR is going to land and do its gene editing. But there's a mathematical likelihood, albeit very rare, that for any 20-letter stretch of that 4-letter alphabet, yes, it occurs in the gene you want to target, but 19 of those 20 letters might arise way over on the other side of the nucleus on a different chromosome in a completely unrelated gene, or maybe not even in a gene at all, just in some random piece of DNA. But the CRISPR machinery is still going to see that sequence and say, aha, there's a sequence I'm programmed to cut, so I'm going to go there as well. So that would be an example of an off-target effect. And if that happens in the cell that you're trying to edit, then you get a little concerned. So there's an art to this CRISPR science in finding the stretch in the gene that you want to edit, where the chances of that happening are essentially as close to zero as you can get. And I think Vertex did a good job of persuading the FDA that those were exactly the sorts of checks that they had undertaken. And so far, in all of the dozens of patients that have been in the trial, I'm not aware of any of these off-target effects being observed. The problems when they've occurred in these sorts of gene therapy trials, more often than not, it's a side effect of the virus that we're using, the Trojan horse that we're using to smuggle these therapeutic genes into the body. And those are things that people are still frantically working on to try to make them as stealthy and as safe as possibly can be, because then we can really open this up to targeting muscle diseases and hopefully brain diseases and a much wider repertoire of genetic diseases.
So you're talking about we're talking about so much finesse in dealing with this. I'm going to kind of jump into the quagmire here. I mean, when we talk about CRISPR, one of the things that's so unique about it is that it's very egalitarian in terms of its accessibility. I mean, I don't even understand. You're going to have to help me get my brain around this. We need to, who gets to decide? That's going to be the big question. But guys are selling kits, CRISPR kits online. Like this bodybuilding guy I found, he's like, you can make your muscles bigger. Like, what is in that kit? I mean, is this guy sending genomes through the mail? I mean, how? I mean, it's when you talk about it, it's clear it takes so much scientific knowledge and so much specialization and so much finesse. But now this it's in the hands of a guy that's just shipping this stuff up at UPS. What is in his kit?
Yeah, yeah. I think I know who you're referring to, but I haven't looked into the details of his kit, and certainly I would not endorse or wish to partake of. I mean, I could use some more. I could use some help in the
Oh,
muscle
you look
building
great.
department, but but I wouldn't be doing it by somebody selling kits, from his basement or whatever. There
But
are
does he have genetic matter in this kit? I mean, don't you need the genetic matter? I mean, don't you need those genomes to inject into your body?
there are genes that there's one specific gene called myostatin that can, when you mutate it, can have a very dramatic effect on musculature. You see this in some animals that have naturally occurring mutations in this particular gene. The regulation of this is an issue, and I think if there was really any I mean, for the most part, I think these sorts of characters have escaped detection, but one would like to think the FDA and FTC are paying a close look. I think the biggest, from the ethical standpoint, the biggest red flag and concern that certainly I wrote about in Editing Humanity, and was just at a meeting earlier this week in DC on this, is about designing using CRISPR on human embryos. Because if you were to use CRISPR to edit a gene in a single cell human embryo, then that edit, now you're playing God, because that gene edit now, as the embryo grows and then is implanted, and 9 months later becomes, a healthy, hopefully healthy baby boy or girl. Now that edit, in principle, could be present throughout that newborn in every cell, including the germ cells, either the egg cells or sperm when the kid reaches reproductive age. And so that edit now becomes part of the gene pool, and now you can pass it down from generation to generation. All the examples, Jonathan, we've been talking about to date, sickle cell, muscular dystrophy, other disorders, there is no prospect, no chance of those gene therapies entering the germline. So you're just treating, even if you're treating a child, you're just treating that child's affected cells, blood cells, muscle cells, what have you. Those edits aren't, those genes are not going to impact the sperm or eggs.
Because it's targeting something so hyper specific.
Correct. But that all changes when you start playing around with human embryos. And of course, this is what the Chinese scientist He Jiankui famously, scandalously did in 2018, when he announced that he had overseen the birth of two young girls. By definition, of course they're young, Kevin. Two babies, Lulu and Nana, in which he had deliberately tried to edit and kind of screw up a specific gene, the gene that codes for the receptor to the HIV virus. And his goal, his manifest goal, was to make these children HIV resistant, because their father had lingering amounts of HIV. He was a former HIV patient. It was a medically unnecessary procedure. It didn't work in the way that he intended. There were credible allegations that he played very fast and loose with the regulations and ethics reviews and so on. And he ultimately ended up serving almost 3 years in Chinese prison when the authorities threw the book at him, in the face of worldwide condemnation, not from religious leaders, from scientists, from bioethicists, and from the public. But it's interesting. He's now back out of prison, back in a lab, wearing his lab coat, and promising. It's almost, I don't know if he's threatening or promising, but saying in very stark terms on social media that he's back. He wants to be the leader of hereditary genome editing, and he thinks he'll start with Alzheimer's disease. And it remains to be, for him to be doing this in full public view kind of suggests that the Chinese authorities they seem to be okay with this. No, they're not muzzling him. They're not putting him. They don't seem to be reprimanding him or warning him. And it's probably only a matter of time before he gets some funding to launch a clinic somewhere to actually try to do this and dupe some people out of some money because they think, okay, well, I know I have an Alzheimer's gene in my family. I'm going to trust this guy. And this guy's a physicist by training. Remember how we said, and you said yourself, how democratized this technology has been. This guy really has no molecular genetic bona fides. He's not trained in this field, and yet he appointed himself to be the beacon of this next generation um, of genetic genome engineers. And so this has people quite alarmed, I think,
Yeah,
because
it's not a big step to Joseph Mengele from there. I mean, you can have some guy in a hollowed out volcano that's Dr. Evil, basically, that's decided he could. I mean, you could literally. I mean, it's a nightmare scenario. You could infect the entire gene pool with, right? I mean, you could effectively
I,
change
yeah, I'm not
evolution.
too worried about those scenarios. I mean, you can only do this one embryo at a time, and so the impact in terms of the human gene pool would take a lot of time to model. But it gets to the ethical question of who among us has the right to start engineering the gene pool of not just the person, the embryo that you're creating, but their children and their children. How do you even model how safe it would be in future generations? Um, there's the latest science suggests that trying to do genome editing, just on a technical level, trying to do gene editing in an embryo, which by definition is a completely undifferentiated kind of naked cell that where literally it will give rise to all the cells, all the multiple different cell types in a adult body. So the DNA is sort of fragile. It's exposed. It's not yet developed all these specialized functions, which makes it would appear much more susceptible to the overreach of CRISPR or whatever gene editor you're using. So when the leading human embryology labs have tried to do this, they've reported major problems, major kind of scarring, rearrangements of the DNA. And it's actually becomes, to my mind, almost a miracle that Lulu and Nana, the Chinese twins born in 2018, it's a miracle they were born, and we think they're healthy, but their identities remain a complete secret, known only to presumably the He Jiankui and a couple of associates. On Twitter, he was saying he wanted to launch a GoFundMe or a foundation based in Singapore to raise money. But I wanted to scream at him, "You broke it. You fix it. This was all you're doing. And now you want to get money from the rest of the world to help fix any potential health problems or cancer scares that these poor children, they didn't ask for this." Um, and I fear that he remains utterly defiant, unapologetic, and he seems to be itching to want to do this again. And
My understanding is that his originally embryonic research was not even that effective. It was like one quarter. I remember that 89, 82 embryos he did this on, and he only had success in 20 something percent of them.
I
He
think
seems a little seems a little overconfident, perhaps.
yeah. Well, there was definitely problems, and when you do CRISPR in an embryo, yeah, he did he did the experiment was doomed to begin with because he was trying to engineer a naturally occurring mutation. We know that millions of people are HIV resistant because they carry a specific deletion of a 32 base stretch in this HIV receptor gene. But to try to replicate that using CRISPR was beyond, I mean, no scientist, not even the Nobel laureates who won the prize in 2020 for CRISPR, could design this precise or mimic this this specific 32 base deletions. So Lulu and Nana are walking human experiments. I think that's what really bothers me the most is they're walking around with different genetic mutations in that gene that have never been modeled, never been studied. And He Jiankui thought doesn't seem to care. He seemed to think he had the right to meddle with their genetic architecture, and we just hope that they will not suffer any long-term health diseases as a result of that.
They had no choice, too. That they're complying against their wills, or
There's like one. They had no choice. Their parents, interestingly, wrote a letter to the court when He Jiankui was being sentenced. And I'm paraphrasing here. This was a letter that was obtained by an academic scientist who's followed this case closely, Ben Hulbert, who's at the University of Arizona, who said, the parents wrote, yes, we did feel somewhat coerced to partake in this CRISPR trial, but it wasn't because the scientists put pressure on us. It was because society put pressure on us as HIV carriers. That's just how China views people with HIV. And so they were persuaded that they had to do something in order to give their children what they thought would be their best and healthiest possible future. So there is another side to the story that hasn't received quite as much airplay.
Okay, I don't mean to go all doomsday on you, but humanity doesn't have a great track record with these types of cutting edge technologies, and I'm looking right at the military industrial complex. I mean, obviously the possibilities for some kind of gene warfare here. Should we be worried about skunk works? I mean, every time humanity gets its, hands on some kind of new technology, it seems like the military is always sort of at the cutting edge, and they almost manage to sort of monopolize it in some way.
Yeah. I'm not too worried about bioweaponry, although this was raised in the very early years of the CRISPR breakthrough in congressional hearings in Congress. So the senators were aware of this. We are taking many of us are taking multiple vaccine shots every winter to ward off against a host of viral threats. I figure we've got plenty of naturally occurring viruses that are very capable of hurting us and killing us if we don't take precautions. We don't really have to worry too much about man-made threats. But then we think back to COVID and the whole raging debate about was it a leak from a lab, and if it was a leak, was it potentially a man-made variant of the virus? And there's very polar schools of thought on that. So, regardless of having CRISPR, scientists already had the ability to artificially synthesize microbial genomes. We've had that. You don't need clever, fancy gene editing tools to do that necessarily. So that threat is with us, and we just have to be vigilant and take care of the threats that we do face.
Okay, so I'm gonna, I don't want to use the word whimsical here because this is science, this is serious, but since it is such an accessible technology, and I know people are already doing stuff with it, like this guy that's selling the kits to try to, increase bicep size or whatever. I got a little list of stuff that I thought touched on different elements of potential uses for CRISPR, and maybe you could just comment on each one. I heard something about glow in the dark plants.
I mean,
Somebody's saying jellyfish DNA into plants, and
we don't need CRISPR for that. That's another Nobel Prize discovery, this green fluorescent protein. So I've seen, every few every few months it seems that the major science journals will put, a glowing mouse or a glowing this or that on the cover. So yeah, we have that technology. Not a threat, not a problem. I don't think anyone's busy trying to make glow in the dark plants for any
Okay, so that's a
obvious.
biohack, but that's not specific to
Right.
CRISPR. Okay, and what about, creating human superpowers? I mean, like, in a sense, like, this guy's like, "Man, if I pump myself full enough of this, maybe I can be the Incredible Hulk." I know this sounds a little but, what are the po
We touched on embryo editing, and so part of the ethical debate has been, okay, it's one thing to correct a genetic disease. We all understand and would agree, virtually everyone would agree that that's a good use of CRISPR. But what about if we wanted to enhance human um, powers in some way? We talked about physical strength, but what about mental strength? What about intelligence? What if we could give kids um, the next generation, the gene for perfect pitch, if we could even get our hands on what that gene actually looks like? And I think that's a whole other program as to whether we should or could or would want to do that. For the most part, if you take intelligence or even longevity, which is another hot topic, there are so many genes involved that I don't think most responsible scientists would even know where to begin. So if a clinic suddenly um, goes online in Southeast Asia or South America or wherever it might be, out of the clutches of the international jurisdiction, and says, "Hey, we can make your child live to 120," I would be very, very cautious because we just haven't modeled that with anything like the kinds of success to even begin to know where to start. I know there's lots of very well-financed groups and many billionaires in Silicon Valley desperately trying to look for what some of these
Yeah,
solutions
well,
might be.
longevity would involve a lot of different genome sequences, right? It's
Exactly.
not one thing like a sig Okay, well, this is making me feel better actually because it's not you can't just throw CRISPR in there and let it, I mean, when you're talking about a quality like, longevity with life or strength, it's gonna affect a lot more things than the one gene
That's
in
right.
sickle cell anemia.
That's
Okay,
right.
so I read about George Church. Now, this is the guy that he's trying to basically de-extinct the woolly mammoth. What can you tell me about how this unfolded?
Yeah. Well, George has been a pioneer in genetics for decades. He's a professor at Harvard Medical School. Has founded dozens of companies, and the one that you're referring to, Jonathan, is called Colossal Biosciences. I think he would have taken the name Mammoth Biosciences, but Mammoth was already taken by one of Jennifer Doudna's companies. So Colossal it is. Um, he was profiled on 60 Minutes a few years ago, and I interviewed him for Editing Humanity. The idea is if we could recreate woolly mammoths and put them back in Siberia, for example, not in some sort of Jurassic Park style theme park, but for sound ecological reasons, keep the permafrost tapped down to stop all of the gigatons of methane that are just frozen, just or locked just under the surface. If that leaches out into the atmosphere, then what we see now around us as signs of global warming will be a walk in the park compared to the future that we'll now face. That would seem to suggest we would need an awful lot of woolly mammoths to even have that sort of an effect. But that's George's vision, and whether he's right or wrong, I love the fact that there are scientists out there who aren't afraid to dream big, and as long as they're doing it responsibly. So just in the last couple of weeks, there have been headlines about how Colossal have gone a small, literally a very small step towards this goal by creating woolly mice. So there are many parts of it. So the idea of creating a woolly mammoth is we're not going to de-extinct. We're not going to sort of do a Frankenstein experiment, but we're going to do a genetic experiment. We're going to take the genome of the extinct mammoth, which we can recreate from all of the DNA samples that are beautifully preserved around the world from these remains, and then we'll line it up with the living genome of its nearest relative, which would be the Asian elephant. And then we can sort of see, and the scientists can compute, where are the key genes that if we took the mammoth gene and put it onto the Asian elephant genetic background, what are the few dozen genes that would get us maybe 80 or 90% of the way towards creating an animal now that would, to all intents and purposes, have the size and perhaps behavior of a woolly mammoth more than an Asian elephant. So there, I think, at least on paper, those sort of gene networks have been described, and fixing sort of the hair growth aspect in mice was just one small step along the way to potentially getting us there. That's probably a decade, we have a long way to go. And their behavioral and sort of, the way that these animals would be reared and not rejected by their, obviously they would have to be born to an Asian elephant family who might look askance when they see this strange creature coming out. So there's going to be a lot of other issues beyond genetic technology to have to sort out. But that's one of the dreams and one of the more interesting and exciting goals of CRISPR in the future.
Now that this is maybe more more a matter of gene splicing or something, but there are UFO conspiracists out there who believe that below the mountains in the Rockies they have the centaur and the minotaur, that these Greek gods, these mythological gods, were actually created by splicing human and animal genes. I mean, yes, I know this is goofy, but you can't help but think about the possibilities when we're talking about being able to edit genetic. Now, I don't know how complex that is. I don't know if there's just one gene sequence for hoofs instead of toes, or I don't know how it works, but now Kevin's going cross-eyed.
Well, yeah. I think, as you said, I think you said it very, very nicely a few minutes ago, Jonathan. With a disease like sickle cell, it's caused by a mutation in a single gene, and we can either target that gene or we can target the regulatory pathway that we know leads to that gene being switched on. So in those sorts of scenarios, we've got literally decades of fantastic scientific research and scholarship that has told us exactly what that pathway looks like. And so we can, with 99.9% confidence, we know where to intervene. Creating sci-fi hybrids, it's fun. It's fun podcast fodder, but not something I'm too concerned about just yet.
Yeah, but a larger question here is how are we going to regulate this on an international level? As you can see, China has different policies than the US. This is something that it seems to me, just in terms of bioethics, needs to be internationally, there needs to be a consensus.
There does, and there is a pretty large consensus that we shouldn't be jumping into embryo editing, but that doesn't necessarily mean it's going to hold. So one thought is, let's de-incentivize the scientists who might want to do this. Let's ask all the editors of Nature, Science, and Cell, and New England Journal of Medicine. We will never publish any work by any scientist who's trying to go down this slippery slope. And the editors of those journals may very well say that's a good idea. We'll do that. But there's a X thousand number of medical journals, and there's going to be one in somewhere around the world that is legitimate enough where the editor-in-chief is, seeking his or her own 15 minutes of fame to say, you know what, I'll take that paper. I don't care if there's pushback. If the work is done and reported, then I'll do that. So yeah, there isn't an international, Interpol's not going to get involved here. And if the Chinese authorities say, you know what, Professor He, we threw the book at you the first time you tried to do this, but it's now 10 years later. The times have changed. China stands for its own ethical policies, whatever those might be, and we don't feel the need to kowtow to whatever the senior scientists in the United States seem to be wanting to tell us we should or should not do. So that's where I see a breach happening. It's possible that there are enough wealthy entrepreneurs who will potentially join forces with one of these scientists to say, we'll set you up in a private clinic where no one can touch you and you can go do your thing. I could see that happening too. So yeah, we have to watch this space.
Well, we're about out of time, but I want to give you just a quick chance to talk towards the future. What area of science or discipline of medicine, what do you believe will be the most transformed by CRISPR in, say, like the next decade or so?
Yeah. Well, I still see a huge upside for treating many more genetic diseases. That's really what the scientists who developed CRISPR primarily had in mind. But when you ask Jennifer Doudna, the UC Berkeley Nobel laureate who co-developed CRISPR gene editing, she will often in media interviews say, actually, it's agriculture where we see more upside, because unless we do something to give plants the genetic resistance to combat drought, pests, viruses, parasites, we're going to lose our food supply very quickly. So in one of the latter chapters of Editing Humanity, I spent time going through many of these examples, the threats against the citrus crop or bananas, and these may sound kind of trivial and
No, it's that.
but
Yeah.
this is important stuff. This isn't just sort of people's economic livelihood. This is potentially existential threats to many of the foods we and millions of people around the world live and rely on. So that requires governments having balancing the understandable consumer public concerns about genetically modified foods. We understand those concerns, but CRISPR is such a much more subtle engineering technique that we're not taking genes from insects and throwing them into the food supply. So we're just trying to fast forward evolution. So instead of spending generations and generations crossing plants to get to the one plant 30 years from now that is resistant to parasite X, we can do it in a lab in a matter of weeks or months. And why wouldn't we then use the technology that we have to help humankind?
Let's save the world with CRISPR. He is Kevin Davis. He is brilliant. Thank you for your patience in walking this dummy through this. It's heady stuff. It's really, Kevin is the author of Editing Humanity, the CRISPR Revolution, and the New Era of Genome Editing. Kevin, thanks so much for joining us today.
Thank you, Jonathan. My pleasure.
A Fresh Face in Hell with Jonathan Evison is brought to you by Fresh Face Pods and produced by Jason Botkin and yours truly. Check us out on Instagram, YouTube, or at our website at freshfacepods.tv. Don't forget to subscribe to our newsletter for updates and special content. And if you love the podcast, by all means, run out and buy all of Jonathan Evison's award-winning novels wherever books are sold.